Dear students, in previous class, you know about the C3 cycle. You know about the C3 cycle. That C3 cycle is also called Calvin cycle. That Calvin cycle was discovered by three members: Melvin Calvin, Benson, and Basham. So those three members are recognized that Calvin cycle. So it is also called C3 cycle. Means the first formed compound is the phosphoglyceric acid. That phosphoglyceric acid having the three carbons. Hence this cycle is called C3 cycle. C3 cycle. And those plants who are follow that this type of cycle, those are called C3 plants. So in this one there are three important steps. One is called carboxylation. Another one is the reduction. So next one is regeneration. So regeneration of the RUBP, because so in that first step, that carbon dioxide is combined with RUBP, that is the primary acceptor of carbon dioxide, RUBP, to form into 3-phosphoglyceric acid, it having three carbons. In that second step, so that 3-PGA is converting into G3P, glyceraldehyde 3-phosphate. It is also called phosphoglyceraldehyde. So, and also in this one, so last step, regeneration of the RUBP is necessary to accepting the carbon dioxide from atmosphere. So in this one, for one glucose, in previous class we have discussed this one. For one glucose, 18 ATP and 12 NADPH. Assimilatory power. The type of assimilatory power is required to synthesis of the one glucose molecule. So glucose, you know very well that C6H12O6. For 6 carbons, 18 ATP, 12 NADPH2 in C3 cycle. So but for one carbon dioxide, how much energy is required? For one carbon dioxide, 3 ATP plus 2 NADPH. Assimilatory power is required to fixation of the one carbon during the dark reaction, means Calvin cycle, and also it is called C3 cycle. So next we are going to the topic about that another topic. So that is called C4 pathway. So C4 pathway are also called H-slack pathway. In previously we are discussing about the Calvin cycle. So that is C3 cycle. In this one, that first formed compound is the phosphoglyceric acid. That phosphoglyceric acid containing 3 carbon atoms. That's why that cycle is called C3 cycle. And also those plants are called C3 plants. So most of the plants are C3 plants. They are undergoes to photosynthesis in Calvin cycle. But another type of cycle is also discovered by the important two scientists. So one of that is H and Slack. So they were worked on the one of that plant that is called sugarcane. The sugarcane scientific name is Saccharum. Saccharum officinarum. Saccharum officinarum, sugarcane, and they discovered that some of that plants are called C4 plants. So in that C4 plants, during this carbon fixation, not only C3 compound, and sometimes they produce the C4 compound also formed during that carbon fixation. So they were discovered, H and slag. They were first time discovered that not only C3 compounds are formed during the carbon fixation, so sometimes they are also fixed, the C4 carbon containing compounds are formed during that photosynthetic carbon fixation. So the type of cycle is called the H-slag cycle. They were worked on the sugarcane and discovered that another type of cycle. That cycle is called H-slag cycle, or also called C4 pathway. So the type of C4 pathway are followed by nearly, so most of the monocots. So most of the monocots are follow the C4 pathway, and also nearly 19 families. So 19 families of the dicot plants, they were also follow that C4 pathway. So how you can say that C4 and C3? C3 means that first formed compound is the phosphoglyceric acid. That phosphoglyceric acid having 3 carbons. That's why that is called C3 cycle. When we are observing in C4 pathway, the primary acceptor of the CO2 is phosphoenol pyruvic acid. So that is combined with carbon dioxide to form into first compound is the oxaloacetic acid. That oxaloacetic acid having 4 carbons. The first formed compound is called oxaloacetic acid in the presence of enzyme PAPase. That means phosphoenol carboxylase, phosphoenol pyruvic carboxylase. So that first formed compound is the oxaloacetic acid. That oxaloacetic acid having 4 carbons, hence this cycle is called C4 cycle. That pathway is said to be C4 pathway. Those plants who are follows this type of pathway, those plants are called C4 plants. When we are observing that C4 plants, there is a specialized structure. So those C4 plants can be show some of the special features in their photosynthetic carbon reduction, especially in their leaves. So you can see the internal structure of the C4 plant leaf. So when we are observing that leaf structure, so upper epidermis, this is the upper epidermis covered with cuticle. Next, this is lower epidermis. That lower epidermis is also covered with cuticle. With stomata, small pores are present. Those pores are called stomata. So, in this one, that between the tissue present in between upper epidermis and lower epidermis, that cells in that midvein region containing xylem towards upper side, phloem towards lower side. It is covered with one of the parenchymatous bundle shape. So that remaining cells are called mesophyll cells. In most of the monocots, that mesophyll is made up of spongy parenchyma. So in most of the monocot tissue, the palisade parenchyma, the spongy parenchyma is present within the mesophyll cells. That mesophyll cells containing spongy parenchyma. That spongy parenchyma are also having, that spongy tissue are also containing the chloroplast. It is also containing chloroplast. So that xylem and phloem, this part is called vascular bundle. That vascular bundle is covered with one of the parenchymatous bundle shape. So this is the parenchymatous bundle shape. So this type of leaf anatomy present in C4 plants. that C4 plant leaves can be show three important specialized anatomical structures within the C4 plants or C4 leaves. So what are those special anatomical features? Those are present within the C4 plants. And you can choose. So the main important one, first one is the cranch anatomy. Cranch anatomy. So what is the meaning of cranch anatomy? So that means cranch anatomy means the internal structure of the leaf can be shown: upper epidermis, lower epidermis. These are the mesophyll cells. Mesophyll cells, that mesophyll cells containing spongy parenchyma, that spongy parenchyma having the chloroplast. So then after, what is this one? So cranch anatomy. So when we are observing that vascular bundle, that vascular bundle is covered with parenchymatous bundle sheath. The vascular bundle is covered with parenchymatous bundle sheath. So that parenchymatous bundle sheath, is looks like wreath. Wreath means ring-like structure. That wreath means ring-like structure. So ring-like structure is present. So that type of anatomy is called cranch anatomy. That cranch is a German word. So it means wreath. Wreath means ring-like structure. So that ring-like wreath is present. So that parenchymatous bundle sheath present around the anatomy of the C4 leaf. That is called cranch anatomy. Once again, you can see the cranch anatomy means each vascular bundle is covered with parenchymatous bundle sheath. Those bundle sheath cells are large in size. Those cells are arranged around the vascular bundle, just like wreath or maybe ring-like structure. The type of special anatomy is called cranch anatomy. That is the specialized unique feature in C4 plants or C4 leaves. And another second one. So when you are observing that second point, so what is the special feature of the cranch anatomy? You can see this another one. So division of labor. So division of labor means that carbon fixation. So that carbon fixation takes place in two types of cells. Carbon fixation takes place in two types of cells. What are the two types of cells? So what are the two types of cells? You can see one by one. First one is the mesophyll cells. So mesophyll cells, and second one bundle sheath cells. So mesophyll cells, and another one is the bundle sheath cells. So that first carbon fixation. So two types of cells are participating into the carbon fixation. So that first carbon fixation means that C4 pathway takes place within the mesophyll cells. So those mesophyll cells lo C4 pathway jarugutundi. So that C4 pathway is also called H-slack pathway. H-slack pathway. The type of C4 pathway takes place within the mesophyll cells. And also another type of cells are also participating to the carbon fixation, especially in C4 plants. Emity and that bundle sheath cells are also participate to fix the carbon through C3 pathway. So that C3 pathway takes place within the bundle sheath cells. That C3 pathway is also called Calvin cycle. So that Calvin cycle or C3 pathway, that is takes place within the bundle sheath cells. That is takes place within the bundle sheath cell. So that is the C3 pathway. So that carbon fixation takes place in two types of cells. One is called mesophyll cells and another one is the bundle sheath cells. In mesophyll cells, C4 pathway takes place, but in bundle sheath cells, C3 pathway takes place within the bundle sheath cells. C4 pathway, mesophyll cells. C3 pathway in bundle sheath cells. C3 pathway takes place within the bundle sheath cells. So, and third important feature, so third special feature in C4 plants, that is the third point, that is the chloroplast dimorphism. Chloroplast dimorphism. Chloroplast dimorphism means presence of two types of chloroplasts within the C4 plants. So what are those two types of chloroplasts? So some of the chloroplasts, those chloroplasts are having grana. So some chloroplasts are containing grana. Those are called granule chloroplasts. Those are called granule chloroplasts. But these granule chloroplasts are present within the mesophyll cells. Those granule chloroplasts are present within the mesophyll cells. So these mesophyll cells are containing granule chloroplasts. And those chloroplasts are present. Those chloroplasts are having the grana. And also, those bundle sheath cells are also present. In that bundle sheath cells, chloroplasts are present, but in that chloroplast, grana are absent. So chloroplasts untai, but grana absent. That's why agranule chloroplasts are present within the bundle sheath cells. So mesophyll cells, granule chloroplasts. So granule chloroplasts are present within the mesophyll cells. Agranule chloroplasts are present within the agranule chloroplasts. Agranule chloroplasts are present within the bundle sheath cells. So that is nothing but division of labor. And that carbon fixation, two carbon fixation within the two types of cells and two types of chloroplasts. So these are the some of the special features present only in C4 plants. So it was first discovered by H and Slack in sugarcane. They were worked on the sugarcane. Most of the monocots are undergoes to C4 pathway for their carbon fixation when compared with C3 plants. So next, and one of the scientists, he was famous Indian scientist. His name Professor V.S. Ramadas. He was also belongs to our AP. So he was the former, he was botany professor in Venkateswara University. His name was Professor V.S. Ramadas. He was also extensively worked on C4 plants, especially in our country. He was a famous Indian scientist worked on the C4 plants. So now itself we are going to the mechanism of the C4 pathway with the help of flowchart. With the help of the how many types of cells? Two types of cells, mesophyll cells and bundle sheath cells. So now itself we can see, we can go to the mechanism of the C4 pathway with the help of two types of cells. So you are going to the discussing about how that two types of cells are participate into the photosynthetic carbon fixation, especially in the C4 plants with the help of two types of cells. Imagine this one. So these are the two types of cells. So one cell is the mesophyll cell and another one is bundle sheath cell. So this is the mesophyll cell. This one is the bundle sheath cell. bundle sheath cell. So first of all, the primary acceptor of the carbon dioxide. So what are the events? Those are takes place within the mesophyll cells. You can see that one. So the primary acceptor of the carbon dioxide in C4 pathway is phosphoenol pyruvic acid. That phosphoenol pyruvic acid can accepting the carbon dioxide from to the atmosphere, but that carbon dioxide is dissolved in water. That carbon dioxide is dissolved in water to form into bicarbonate. So that bicarbonate, so that C4 plants most important MSET bit. So that is those C4 plants are absorbing the carbon dioxide in the form of bicarbonate. 2016 MSET bit. Only this line that is not available in textbook line, but it is available in the diagram. So when we are observing the diagram in your textbook, there is a small bicarbonate is available. So that is accepting by that carbon dioxide is accepting by phosphoenol pyruvic acid. That is the primary acceptor of carbon dioxide. To first formed compound is called oxaloacetic acid. That first formed compound is oxaloacetic acid. So that oxaloacetic acid having 4 carbons. That oxaloacetic acid having the 4 carbons. Hence this pathway is called C4 pathway. Those plants are called C4 plants. Those plants are said to be C4 plants. That pathway is called C4 pathway. In the presence of enzyme, one of the most important enzyme, that is called peptase. Peptase means phosphoenol pyruvic carboxylase. So simply that is abbreviating as peptase. So this is the first step in C4 pathway. Then after this oxaloacetic acid, by utilizing the assimilatory power like NADPH, that utilizing the assimilatory power like NADH2, so it can be utilized only that hydrogens present within the NADPH, that is converting into NADP plus. It's converting into another 4 carbon containing acid. That another 4 carbon containing acid is malic acid. So it is converting into another 4 carbon containing compound, that is called malic acid. In the presence of enzyme malic dehydrogenase. In the presence of enzyme malic dehydrogenase. So it is oxaloacetic acid is converting into malic acid in the presence of enzyme malic dehydrogenase. That assimilatory power is used in this reaction. So that is NADPH that was synthesized during the light reaction. In previous calls we are also discussing about the NADH and NADPH, nicotinamide adenine dinucleotide phosphate. So then after that malic acid is enter into the another type of cells. Those cells are mesophyll cells. So that is enter into the bundle sheath cells. So that is malic acid. So that malic acid is enter into bundle sheath cells for the remaining reactions of the C4 cycle. But in sometimes that malic acid is undergoes to the transamination and producing into aspartic acid. Sometimes are also that malic acid is undergoes to transamination that producing that is converting into aspartic acid. So based on that one, some plants are said to be aspartate forms and some plants are said to be malate forms. Based on the remaining reactions, those are present within the C4 plants. So C4 plant lo జరిగేదాన్ని బట్టి malic acid తో కంటిన్యూషన్ అయిందనుకోండి, సో దట్ టైప్ ఆఫ్ సైకిల్ ఇస్ ద malate form. ఒకవేళ maybe malic acid is undergoes to transamination, that is converting into aspartic acid. That aspartic acid is enter into the remaining reactions of the remaining reactions of the C4 plants. So such a time, so that is called aspartate forms. But on that board, you can see the malate form. So that malic acid is entered into the bundle sheath cells. What happened within the bundle sheath cells? That malic acid is undergoes to oxidative decarboxylation. Undergoes to the oxidative decarboxylation. So oxidative decarboxylation means oxidation means loss of two hydrogens. Those hydrogens are accepting by to the NADP plus, that is reduced into NADPH. You can see this one. One of the NADPH was synthesized within the bundle sheath cells, but one of the NADPH is utilized within the mesophyll cells. Both are compensated with each other. That's why there is no need not any additional NADPH during that C4 pathway or C4 cycle. ఈ C4 ప్లాంట్స్లో చూస్తే మనకి ఇంకా అడిషనల్గా NADPH అవసరం లేదు. ఎందుకు అంటే బికాజ్ వన్ ఆఫ్ ద NADPH ఈజ్ సింథసైజ్డ్ విత్ ఇన్ ద బండిల్ షీత్ సెల్స్ and another NADPH was utilized in mesophyll cells. That's why both are compensate with each other. So both are the compensate with each other. That's why there is no extra NADPH not required in C4 cycle. Next, oxidative decarboxylation. What is the meaning of decarboxylation? Loss of carbon dioxide. Hence that malic acid is converting into pyruvic acid. Malic acid is converting into pyruvic acid. So that is also 3 carbon containing compound. It is the 4 carbon containing. Malic acid is the 4 carbon, but 1 carbon can be lost in the form of carbon dioxide. So that pyruvic acid. So immediately that releasing carbon dioxide is accepting by to the, already I told that one. How many carbon fixations? 2 carbon fixation. C4 pathway takes place in mesophyll cells. Now itself that CO2 is accepting by to the RUBP, that is converting into phosphoglyceric acid, that is also regenerate, and some of the phosphoglyceric acid. So some of the phosphoglyceric acid is helpful to the synthesis of the glucose through the C3 pathway. So that C3 pathway takes place within the bundle sheath cells. So C4 pathway takes place in the mesophyll cells. So this is the first carbon fixation. That carbon dioxide bicarbonate is accepting by to the phosphoenol pyruvic acid. So that is the primary acceptor of the CO2 and producing into oxaloacetic acid in the presence of enzyme peptidase. Okay, that is the first carbon fixation. That carbon fixation is the C4 pathway. And the second carbon fixation, so C3 pathway. That C3 pathway now itself, that C3 pathway is also called Calvin cycle. That Calvin cycle takes place within the bundle sheath cells. That Calvin cycle takes place within bundle sheath cells. So that food material also synthesized through the Calvin cycle. So food is also synthesized within the bundle sheath. Food does not synthesized within the mesophyll cells. So that starch synthesis takes place within the bundle sheath cells, because in that bundle sheath cells agranule chloroplasts are present, only stroma is present. In that stroma, synthesis of the food material takes place within the stroma. Okay, after onwards, that pyruvic acid is reback into the mesophyll cells. Already the food was synthesized within the bundle sheath cells through the Calvin cycle or C3 pathway in the presence of enzyme. You know very well that enzyme is called Rubisco. That Rubisco enzyme is mainly involved in C3 cycle. So in that C3 cycle or Calvin cycle, food was synthesized within the, only that food is synthesized within the only bundle sheath cells. Okay, so after onwards, that pyruvic acid, that 3 carbon containing pyruvic acid is reback into the mesophyll cells. That pyruvic acid, so that pyruvic acid is enter into reback into the mesophyll cells. But then after that pyruvic acid, by utilizing the 2-ATP, so that pyruvic acid by utilizing 2-ATP and producing into PAP means phosphoenol pyruvic acid. So phosphoenol pyruvic acid by utilizing the 2-ATP, by utilizing 2-ATP, so that converting into 2-ADP plus 1-PI. So another PI is used to the one phosphate is attached to the pyruvic acid, hence that is called phosphoenol pyruvic acid. Another phosphate is also released for their energy purpose. They utilize 2-ATP molecules in the presence of enzyme pyruvic kinase. In the presence of enzyme pyruvic kinase. Remembering that one, ekkadaina ATP vachindi ante akkada kachithanga unde enzyme enti ante kinase. It may be NADPH or NADH2. So NADH2 involvement undanuko enzyme eppudu kuda dehydrogenase. So enzyme ekkudu kuda dehydrogenase vastu untundi. So with the help of enzyme pyruvic kinase, so it is converting into phosphoenol pyruvic acid. So this is the entire C4 pathway. So that carbon fixation, 2 carbon fixations takes place within the C4 plants. First carbon fixation in mesophyll cells, second carbon fixation in bundle sheath cells. First carbon fixation that producing the 4 carbon containing compound oxaloacetic acid. Then after converts into malic acid. Malic acid enter into the bundle sheath cells. Within that bundle sheath cells, that is converting into within that one that is converting into, so that is undergoes to oxidative decarboxylation. In that oxidative process, they produce the NADPH. Decarboxylation means loss of carbon dioxide. That carbon dioxide is enter into that carbon dioxide is accepting by RUBP, ribulose 1,5-bisphosphate in C3 cycle, and synthesis of the food materials takes place within the bundle sheath with the help of C3 pathway. That's why C4 pathway takes place in mesophyll cells. C3 pathway takes place in bundle sheath cell. Then after that pyruvic acid is reback into the mesophyll cells. Pyruvic acid reback into mesophyll cells. So in that mesophyll cells, that pyruvic acid is converting into phosphoenol pyruvic acid by utilizing 2-ATP. So during this last step, for every carbon dioxide, additional 2-ATP is required. So now itself we can see how many ATP are required to synthesis of 1-glucose during C4 pathway or C4 cycle, so H-slack cycle. You can see this one. So already C3 cycle is there. So in that C3 cycle, 18 ATP for 1-glucose. So for 1-glucose, 18 ATP and 12 NADPH. Assimilatory power is required for 1-glucose. 1-glucose means C6H12O6. For 1-glucose, so 18 ATP and 12 NADPH through the C3 cycle. But in C4 pathway, so how many NADH2 are additionally required? There is no need of the additional NADPH are required within the C4 pathway because of one NADPH is utilized in this one NADPH is so synthesized in this one. సో బండిల్ షీత్లో ఒక NADPH తయారవుతుంది, బీసోఫిల్లో ఒకటి యూజ్ అవుతుంది. అంటే బోత్ ఆర్ కంపెన్సేట్ విత్ ఈచ్ అదర్. సో దట్స్ వై దెర్ ఈజ్ నో అడిషనల్ NADPH ఆర్ రిక్వైర్డ్. సో అయితే హౌ మెనీ ATP? సో ఫర్ వన్ కార్బన్ డైఆక్సైడ్, so if maybe one carbon dioxide is undergoes to C4 pathway, additional 2 ATP are required. But glucose having how many carbons? 6 carbons. That glucose containing 6 carbons, that's why additional 12 NADPH. So oka carbon kaitenemo 2 ATP. So for one carbon 2 ATP, but so for one carbon 2 ATP, but glucose containing 6 carbons, that's why additional 12 ATP are required. So total number of ATP, can we count that one? So total number of ATP 18 plus 12, total 30 ATP and 12 NADPH. 12 NADPH assimilatory power is required in C4 plants for 1 glucose, in C4 plants for 1 glucose molecule. So that is the additional ATP. So total ATP for 1 glucose and adigar anukondi. So in C4 plants to formation of the 1 glucose molecule, how many ATP are required? 18 plus 12, 30 ATP are required. 30 ATP are required for 1 glucose molecule during the C4 pathway. But in C3 so how many additional ATP? How many additional ATP are required? How many additional ATP are required for the to formation of the 1 glucose in in C4 plants? That is 12 additional ATP are required. 12 additional ATP are required in so last step of the C4 pathway. So total number 30 ATP, 12 NADPH, but additional ATP only 12 ATP. So 12 additional ATP are required during the C4 pathway. So this is the C4 pathway that takes place within the bundle sheath cells and mesophyll cells. So when we are observing in this pathway, this is the most important significant into the product, increasing the productivity of the crops when compared with the C3 and C4 plants. So those C4 plants are highly efficient in C4 plants are highly efficient in rate of photosynthesis when we are compared with C3 plants. So then after you can see there is a short plants are there, short topic. You can go to that CAM plants. A short topic is there, that is called CAM plants. So CAM means crassulacean acid metabolism. Crassulacean acid metabolism. So this type of crassulacean acid metabolism is first discovered within the one of that family. That family is called Crassulaceae. So Crassulaceae are also that family Cactaceae. So Cactaceae are Crassulaceae family. That's why these are called CAM plants. What are the special features in CAM plants? So in this one, to reduce the rate of transpiration, the stomata in those plants, those stomata are closed during the daytime, and those are open during that night time. So this type of special movement of the stomata, that is in previous chapter, scotoactive movement. So best example you know very well, bryophyllum. bryophyllum, and also you know that opuntia, cactus. So in that opuntia, cactus, so in this one the stem is modified into phylloclade. So in that phylloclade, the stem for forming the photosynthesis, all the leaves are modified into spines, the type of cactus, and also bryophyllum, in our Telugu Ranapala. So in this one epiphyllous buds are helps in the vegetative propagation. So ఇలా ఉన్న ఈ ప్లాంట్స్లో చూస్తే those stomata are closed during the daytime and open at night time. So the type of movement is called the scotoactive movement. So these are commonly occur within the Crassulaceae family or Cactaceae family. So that's why, so in that camp plants, that C4 pathway and C3 pathway, the C4 pathway and C3 pathway, both are takes place within the mesophyll cells. Both are takes place within the mesophyll cells. There is no change. Those metabolic activities, all those metabolic activities are equal to that of whatever it may, those are takes place within the C4 plants. Same away jarugutayi. అయితే both are takes place within the mesophyll cells. మరి what is the difference అంటే only time gap. Time gap is the difference. That means C4 pathway takes place during the night time. Night time C4 pathway takes place. In that night time they are producing into more amount of malic acid. That malic acid is preserved within the vacuole. That malic acid is preserved within the vacuole during the night time, and also that food preparation or C3 pathway takes place during the daytime. That is the difference between C4 plants and camp plants. In C4 plants, two types of cells are participate: mesophyll cells, bundle sheath cells. Mesophyll C4 pathway, bundle sheath C3 pathway. But in camp plants, both C4 pathway and C3 pathway, both are takes place within the only single cell, that is called mesophyll cell. That is the mesophyll cell. But there is a time gap is there. So during that night time, those stomata are open during the night time, scotoactive movement. That's why entry of the carbon dioxide takes place only night time. That means C4 pathway takes place during the night time, and so malic acid is formed during night time, that is preserved within the vacuole. So then after that is comes out into the chloroplast and enter into the C3 pathway during that daytime. So that is the camp plants Crassulacean acid metabolism. Okay, so today we are completing about this C4 plants and camp plants, their mechanism. Next, in becoming class, we are discussing the photorespiration and difference between C3 and C4 plants and factors those are affecting the rate of photosynthesis. Okay, thank you very much.